Hi, I'm Len Calabrese from the Cleveland Clinic, and I'm here on Room Now with two experts in immune-related adverse events from cancer immunotherapy. Laura Capelli from Johns Hopkins, Cassie Calabrese, who I actually know, from Cleveland Clinic. And, you know, 4 or 5 years ago, you had to find one or two presentations on this, and by one calculation, there are over 60 abstracts that involved cancer and rheumatic disease at this meeting. So, highlights. Laura, give me a couple.
So I think we had a really great oral abstract session yesterday, and there was some very interesting pharmacovigilance data that was presented from the group from the Sorbonne. So they looked at the World Health Organization database for iRAEs, so immune-related adverse events secondary to immune checkpoint inhibitors, and they found about 600 reported cases of arthritis and about 400 of myositis. But I think the real take-home message from that abstract was that in the patients with myositis, those who would have fatal cases typically developed the first symptoms about 2 weeks before those who would have not fatal cases, which I think is really thought-provoking about the pathogenesis that's going on there. You know, do these people have already
Pre-existing? Oh, absolutely.
Yeah.
Great presentation, and myositis, very serious and got to be picked up early. Cassie, give me one.
Yeah, so many great abstracts. I think I've been told there's 60 some. I'm not sure if that's accurate, but also yesterday at the oral session, something kind of new and different from Marie Costine in Bordeaux looked at the effect of microbiome altering medications on iRAEs, and what they did was a retrospective study, I think 600 some patients, looking at administration either within the month before or month after of a handful of different types of drugs, including antibiotics, steroids, some psychotropic medications like statins, ACE inhibitors, and looked at the effect on overall survival and tumor response. And interestingly, something we've kind of suspected is that prednisone equivalent dose of 10 milligrams or more daily at baseline had negative effects on overall survival and tumor response. Also interestingly, similar negative associations with antibiotics and proton pump inhibitors. That was another one. So I think take-home point, if we can avoid baseline steroids at a dose of 10 milligrams or more daily, that's probably something we should be doing prior to starting immunotherapy.
Yeah, it's a very provocative study. You know, these immune diseases, as Laura mentioned, some may be pre-existing autoimmunity, some may be operating through novel mechanisms of breaches of tolerance, and we have an incomplete understanding of both the genetics, which are a partial contributor, and environmental factors. And the antibiotic story's been out for a long time, but geez, PPIs and these other drugs, and are they operating?
They're so commonly administered to people.
So my actual personal discussion with patients and friends who have undergone this is to try to live as naturally as you can. Eat a healthy diet and do all the type of wellness behaviors you can before doing this, and try to clean your system out. We are at the beginning of this. I think that one more abstract from Laura, and we'll wrap this up.
So I think we're going to go from big, database studies to small. And so the Mayo group presented also at the oral abstract session some flow cytometry data looking at peripheral T cells in patients who developed new inflammatory arthritis from checkpoint inhibitors as compared to healthy controls, RA controls, and checkpoint inhibitor controls who didn't get arthritis. Bottom line is, small number of patients, needs more validation, but intriguing that the immunosenescent phenotype seemed to be up and the exhausted T cells seemed to be down in the checkpoint inhibitor patients, in a similar way actually to the traditional rheumatoid arthritis patients. So I think that it was provocative, small number of patients, good pilot data for more study.
I agree 100%. There's just a recent roundtable discussion, I think in Nature Immunology, taking the world-leading experts on exhausted T cells, and there's really a lack of uniform definition of how to define these by advanced flow and metabolomics and transgenomics. So again, I think it's a great start. It's telling us something, and hopefully we can build upon it with larger studies. I want to thank everybody for coming into Room Now. iRAEs are here to stay. They're only going to get bigger, and next year we're shooting to maybe have our own abstract category.
Fingers crossed.
Yeah.
Thanks.